Hello Matrix and welcome to Was a Matrix. My name is Looney and I'll be assisting you to get all of your questions through to us. Today we're focusing on your exam guide for Life Sciences Paper 2. All you need to do to get through to us is follow Was a Matrix on our social media platforms as well as hit us up on our WhatsApp line. We've got a cool competition going on for you guys, so please do stay tuned for all of those details. I'm not alone. With me are my sign language interpreter, Nicolene, as well as our teacher, Eugen. Over to you guys.
Welcome, grade 12s, to another installment of Life Sciences. We've spent some time over the last week looking at preparing you best for paper 1. Today we're going to spend some time looking at preparing you for Life Sciences paper 2. In this segment, we're going to look at the structure of the exam. We're going to spend some time looking at how to best prepare for paper 2, and also we're going to look at some questions that have challenged learners over the years. And so we're going to focus on those questions and give you an insight as to how to best approach these questions. So let's get straight into the lesson and let's look at the structure of this paper 2. So when we look at the exam guide for paper 1, we looked at the structure of it. Now when we look at exam for paper 2, we know that the examination consists of a 2.5-hour exam, and this exam basically needs to be completed in 3 sections. What do I mean by that? Well, if you recollect, we have section A, B, and C, and these sections are allocated different types of questions. If we look at section A quickly, we would see that section A has a section of short questions that generally require you to read and extract information from. They include objective questions such as multiple choice questions and terminology. We often see questions based on matching columns, true and false statements, etc. And this is the section where you often find the easier questions. And I often refer to these as the lower hanging fruits that you can easily access and score your marks in. When we look at section B, this section is out of a total of 80 marks. There's a variety of questions that come into these questions. Here you probably have 2 questions of 40 marks each that could take you through the second part of this exam. You need to note that each section will be divided into between 2 to 4 sub-questions. Finally, the section that most of you dread, but I assure you it's probably the best question in the paper to attempt. And that is question 4, and that's looking at the essay. So we spent some time last lesson looking at how to attempt an essay. Today we're going to look at how to look at the structure of the exam and to focus on specific questions based on the allocation of the marks per question. Looking at the structure of the exam, we know that DNA contains a significant portion of the paper, and by that I mean 19% of the exam is made up of DNA, which includes sections that you have really done well in during the beginning of the year. We know that meiosis is a section that is in paper 1 and paper 2, so you've done a lot of studying on that section. You need to transfer that knowledge through into your preparation for paper 2. We know that genetics is a significant chunk of this, and we see that it's a 45 contribution mark to that entire 150 marks. And then finally, a section which we all really have enjoyed this year, and that's a total of 66 marks, which makes up almost 50% of the paper. And hence, if you consider the breakdown of these topics, you need to accordingly plan your schedule around the time that you spend focusing on these 2 topics, namely genetics and evolution. Guys, just to overview each aspect in terms of the content required for each topic, I'm going to very quickly give you an overview of that so that you can do a quick checklist when you are preparing for this exam. So DNA, the code of life, consists of the following sub-aspects, and we refer to that as you got to know the structure of DNA, you got to understand the terminology that we've used around nucleic acids, the structure of both DNA and RNA. We've got to know and tabulate the differences between the molecules DNA and RNA. we've got to look at a very important process of replication and its importance and link that to meiosis. We also need to know the significance of DNA profiling and what its uses are. And most importantly, the process of protein synthesis, which is essentially a key component to understanding the process of how proteins are made. As I said, that meiosis you've prepared in paper 1 on this section, so we've got to know the process of meiosis and link that to cell division. We've got to look at the significance of meiosis and when it occurs and what's the importance of having chromosomes numbers halved. We've got to look at the impact of what happens when meiosis goes wrong, so abnormalities such as Down syndrome and other syndromes that do occur because of non-separation during anaphase 2. We also need to look at the differences between meiosis 1 and meiosis 2 as 2 stages of the cell cycle that the cell goes through. We've got to spend a lot of time looking at genetics, and hence it is important that you use this checklist to carefully check through each of these components when you're preparing for paper 2. We've got to know that there's a vast array of terminology that are important. So create that terminology vocab list on elaborate terms that are present on this topic. Understand the types of patterns of inheritance. By that I refer to the pattern of complete dominance, incomplete dominance, and co-dominance, and how those characteristics are inherited. Very importantly, we've got to look at the inheritance of sex-linked characteristics, which I'm going to spend some time looking at this lesson. We also need to look at how sex is determined in an individual. We've got to look at the dihybrid cross as a mechanism of being able to look at how characteristics are inherited together when we look at the inheritance pattern. We also need to spend some time looking at mutations, guys. An important expression of characteristics is what happens when things go wrong during either meiosis or even during DNA replication and protein synthesis. Pedigree charts are an important component of understanding the inheritance of characteristics in a family. More important than genetic engineering is understanding how it is applied. So things like somatic cell nuclear transfer, looking at genetically engineering insulin. We've got to understand that, very important. The use of genetic counseling in terms of as a mechanism to counsel and to offer advice. The use of DNA fingerprinting in paternity tests is an important aspect of being able to apply your understanding. And then we get to a chunk of paper 2, which is evolution. And this makes up, as I said, probably 50% of the paper. We're looking at about 44% of the paper made up of evolution. So we look at evolution as, firstly, the evidence that is needed. We need to look at what creates an opportunity for variation which drives evolution. You've got to spend some time looking at the theories, especially Lamarck's theory and Darwin's theories. So we talk about the theory of natural selection and compare that to artificial selection. We also know that there are terms such as punctuated equilibrium and gradualism, the concept of speciation and how species form through allopatric and sympatric speciation. We need to look at the mechanisms that are involved in that process of reproductive isolation. And finally, we've got to spend some time looking at how evolution has occurred in present situations, and we draw reference to resistance of antibiotics in bacteria. We also need to divide that planning into how we prepare on human evolution, knowing that South Africa and Africa plays a significant role in understanding the evolution of humans. We've got to compare humans to other African apes. We've got to look at the differences between humans and African apes. We've got to spend some time looking at major phases in hominid evolution. The two theories that are important would be the out of Africa hypothesis and the multi-regional hypothesis, which you need to understand and be able to differentiate. And then interpreting and understanding phylogenetic trees. So guys, a good point to start off this would be how to manage your time. And often you would have seen that the paper is an elaborate paper of 150 marks in which you have 2.5 hours to attempt all those questions. What is the strategy that you employ when attempting to answer these questions? here's a recommended amount of time that you should be spending per question. And this is purely based on looking at the level of degree of difficulty per section and the amount of marks that are based per question. So as I mentioned at the start, your question 1 is the section that has your lower hanging fruit. By that I mean the easier questions. And this amounts to 50 marks. So you know that sometimes reading through terminology in multiple choice goes a little quicker than normal. So the recommended time is around 45 minutes for that section. Questions 2 and 3 are 40 marks each, and these should take you approximately 35 minutes each. And having done that, you still have time now to spend time on your essay, which is question 4. For that 20 marks, you should be allocating around 15 to 20 minutes. What is very important is the last bit. Often learners will write till the last minute of the exam. But by looking at this allocation, it gives you 20 minutes which you can use to go back and to reflect on your answers and responses for each of the previous questions that you've answered. And this will allow you to be able to review your answers, to probably add on to them, and to attempt questions that you might have left out. And that's critical to the way you plan this paper. So guys, let's look at how we get into preparing for this paper in the next segment. Luni, over to you.
Thanks, Eugen. Guys, let's take a quick break and we'll see you straight after this. Welcome back from the break, guys. Thank you so much for tuning in. If you've just joined us, we are assisting you with your exam guide and preps for your upcoming exam. If you're someone who's constantly running out of data, then this next competition is just for you. Wazometrics brings you the hashtag WazoWinner competition, where 2 lucky matrics stand a chance to win 2 gigs of data. All you need to do to enter is head on over to our Facebook page and all of the details will be there. Well, enough talking from my side, over to you.
Welcome back, folks. Thank you for sticking in there. In this segment, we're going to look at the best way for you to prepare for paper 2. We've just looked at the structure of the exam. We've looked at the mark allocation. We've also spent some time looking at the specific aspects, or I refer to that as specific topics per section. And I think that's a very important point to start your preparing for this. So how do you prepare for this exam? Let's have a look at that, and we'll discuss a strategy that's going to help you better prepare for your final paper 2. So let's look at the tips for preparing for paper 2. It's important that you firstly revise each section using your summary notes. And at this point in time, you've spent the year writing notes in your notebooks. You've spent some time during the prelims preparing summary notes. Guys, you don't need to recreate the wheel. So go back to your summary notes and read through these. And if it helps, and if you haven't, then try highlighting the key concepts. Underline important words. And as I always mention to the learners, by underlining or by adding color to your notes, you bring life to that. And often we are able to recognize color, we're able to see things that are underlined and recall that. So get into the habit of highlighting key concepts, underlining focus ideas. And if you have to, then write them out on a small piece of paper and use that as a chit reference when you are preparing the night before the exam. It's important that you create a list of terminology for each topic. And by that I mean it's a good checklist for you to check before you get into the exam, either the night before or the morning. So having a list of terms, just listing them. So identify the topic, list all the terms, and you go through that as a checking mechanism. And that allows you to have a good reflection and recollection of the specific terms per section. And once you have that, it helps you to access those terms and those important concepts when you're attempting a question. It's important that you use diagrams and you learn from diagrams that are relevant in this section. Label these diagrams, add annotations to them. What do I mean by the term annotation? Well, often in this section there are diagrams of, in evolution, skulls. There are flow charts like DNA replication. There is protein synthesis, which contain lots of information with different types of structures that have significant roles. So on an A3 sheet of paper or A4, illustrate the images that are important. Use key concepts and words, highlight them, add in the function, describe the structure. And that's one way in which you can look at a topic like DNA replication and understand how the process occurs. Another key way to remember processes is using flow diagrams. And of recent, the examiners have asked, describe the process using a flow diagram. A flow diagram are key phrases that are short, that describe each step, that are indicating the flow of information. So you can use these flow diagrams, especially for processes like protein synthesis, where you have to describe transcription and translation. Illustrate these on a diagram and then use the annotation, simple labels describing the characteristics of each step into a flow diagram. A very effective way of preparing for questions where processes are needed. You can also use the same technique in understanding the process of cloning, somatic cell nuclear transfer, or even in trying to understand the process of genetic engineering and how recombinant plasmids are created. Another effective way is creating timelines. So for the timeline of the hominid evolution, use a timeline to indicate the process of development. Identify key characteristics using terms that are relevant to each section. And that is how a list of terminology as well as labeling diagrams and using flowcharts can enhance the ability for you to learn concepts and recollect them, but most importantly, effectively review these soon before you write. In biology and in life sciences, we look at structure, function, and location. And that is what underpins the learning of concepts in life sciences. So identify a structure, identify its function, and look at how that relates to where its location is. So if we're looking at the process of protein synthesis, identify structures like DNA, mRNA, rRNA, and tRNA. Look at their structures. DNA is double-stranded. RNA would be single-stranded. If you look at the TNA molecule, remember its shape and look at its function. And in so doing, you connect the different concepts and structures into understanding the big picture. And that is how you learn complex processes that involve a series of steps, and you make that much easier for you to recollect in the time of an essay or in the time of a shorter or longer question that has an 8-mark description of a process. Okay. Guys, there's an important resource called the Mind the Gap booklet, and that booklet has a whole list of diagrams. It has tips on how to prepare. It's got questions that are specifically related to concepts that have not been well understood. Refer to that. It's an excellent resource. Tips for preparing for paper 2, and we'll continue with that. Print out a few papers and look at them all. Look at questions specifically pertaining to a topic. Okay? And often I do this. I get the learners at my school to print out the last 3 or 4 year exam papers. You then open them up and you look at all the topics relating to, say for example, DNA protein synthesis. Now that you've spent an hour or two studying a specific topic, attempt to answer these questions without having to look at your notes. Know that the best way to prepare is to physically write down answers. And yes, you might get it wrong, but you have the opportunity to immediately reflect on the right answers and correct them. And that is exactly how learning effectively improves your preparation for this exam. So again, there's no better practice than looking at past exam papers and attempting to write these as an exercise of simulation of an exam. Check these answers using the memo, and that's important because when you do that, you're able to reflect on what you've got right and what you've gone wrong. I often use an old and trusted method called CAR, and this is a method that's based on identifying the content, which I refer to as the aspects of each topic. Identify these first. Use that as a checklist to tick off as you've studied them. Attempt to answer questions, and that's the application of knowledge. Guys, the reason why we set exams and tests is to see how you apply your knowledge. And so if you can incorporate some form of questions, and as I said, from past year exam papers, you are able to apply your understanding. And yes, during the practice of exam papers, you might feel that you are not confident enough because you might be only 50% prepared. That will give you an opportunity to go back and to review the content. So when you review content, you identify questions or sections that you have not understood well. So sections are identified. You then spend some time reviewing that specific topic. You go and look at your terminology list. You go and you check your notes, and then you review that. So if you've not understood the process of protein synthesis, go back to the drawing board, review the process, and then you go back to learning the content and then applying that by attempting another set of questions. And so I call that the CAR model, which is content, application, and then review. And you constantly do that until your confidence level starts changing, and you will see that you start improving. And that is what preparing for an exam looks like. Okay, so guys, we've looked at tips on how to prepare for this exam. In my experience, and having chatted to learners, there are some questions that will always challenge learners. Sections such as crosses, so your genetic crosses, your monohybrid crosses and your dihybrid crosses are generally questions that will appear in every year. So these are guaranteed questions that the examiner are going to set. So how do you prepare for that? Focus your revision on that. You should be able to prepare a question on a monohybrid cross and understand what a dihybrid cross is. So I'm going to attempt to solve a problem on a monohybrid cross and talk you through the method of laying out a monohybrid cross and looking at where the mark allocation is per question or per section of that monohybrid cross. Okay, so let's look at this question. So we're going to look at the process of what complete dominance is, and remember that we have spent some time this year looking at two other patterns of dominance, which would be incomplete dominance as well as co-dominance. And co-dominance and incomplete dominance essentially are different patterns of inheritance, and it's important that you're able to differentiate between the three. In this example, we're looking at an example that talks about specifically complete dominance. So here's the question. In humans, the ability to roll the tongue is due to a dominant allele. And by dominant, we should know the difference between what dominant and recessive means. A man who is heterozygous for this condition, tongue rolling, and I'll explain heterozygous in a bit. Remember that in heterozygous, we're referring to the representation of a dominant allele, and in this case, I'm going to use the letter T, and a recessive allele, the small letter t. So a man who is heterozygous, meaning that he's got the dominant allele and the recessive allele, and a woman who cannot roll her tongue, have children. So when we look at the condition of not being able to do this specific action, would mean that she is recessive for that condition, In other words, she is homozygous recessive, and that would mean that she has 2 recessive alleles for that condition. And I think it's important that when you read a question that you're able to identify and highlight the key components of this, which help you to identify what we call the genotype and the phenotype of the individuals involved in the cross. So the question goes on further, and we'll read through the instructions. Use the symbols capital T and small t for the alleles of tongue rolling characteristics, and represent, we've got to represent a genetic cross, and that's the part of the question that we need to represent, to determine the possible genotypes, which are the genetic expressions, and phenotypes, which is the physical expression of their children. And it's important that the question stops by you looking at the mark allocation. And for me, that is the most important part of the question, is trying to identify the mark allocation. And that often talks to the degree of thoroughness that you need to have for this answer. So let's get straight into how I would lay out this step by step so that I could achieve the full 6 marks for this question. So guys, let's look at how we lay out a genetic cross. And as you've seen, I've inserted on this image the steps on how to lay out a genetic cross. So when solving a genetic cross, it's important that we first use the allocated key. And in this question, they had indicated to us that we need to use the letters capital T and small t. So we're going to use the letters capital T and small t. I'm just going to get back to that slide, and we can refer to this again. So when we look at the genotypes of these individuals, we've got to use the letters capital T and small t. In so doing, the genotypes that were given was that there was a male that was able to roll his tongue, that married a female that was a non-roller. And knowing that being able to roll your tongue is a dominant condition, we know that it mentioned that the individual was a heterozygous individual. So the next part of the diagram is for us to be able to identify the genotypes. A heterozygous individual is one that has the dominant allele as well as the recessive allele, and hence that is something that we must clearly indicate. We also know that the female was a non-roller, meaning that she was homozygous recessive, and hence we've given her 2 recessive alleles. The next step in our diagram is to be able to identify the gametes, and we should indicate the number and the process of how that occurs, and it occurs through meiosis. During meiosis, we know that these gametes separate out, and hence we've indicated these as separate letters, and that's the identification. You notice that I've indicated a single T. Some of you may choose to represent both the T's. It will still give you the correct ratio at the end. For me, I've used a single T in this cross. The next process shows you the process of fertilization, and that can be done using either the Punnett square or using a simple cross diagram as I've illustrated here, where we cross the gametes that produce the possible F1 genotype ratio. And these include an offspring that is heterozygous, as well as an offspring that is homozygous recessive, meaning having 2 small characteristics for this specific characteristic. We end off the equation or the genetic cross showing the ratio of the genotypes, and in this case we have 1 tongue roller, which is that individual, to possibly 1 non-tongue roller. And we say that there is a 50% chance of having a tongue roller compared to a 50% chance of having a non-tongue roller. And guys, so we've spent some time looking at how to lay out a genetic cross. Remember that there are variations to this. Another key factor to understanding how to do genetic crosses is to consider the type of inheritance. By that I mean, is it an autosomal condition or a sex-linked condition, meaning gonosomal? So is it found on the X chromosome? And if it is found on the X chromosome, which are examples such as hemophilia, color blindness, for those examples, we've got to indicate the gender as well as the allele. And hence you would use the letters XX and XY for females and males respectively. And thus, you should also include the gender and the allele of the individuals when doing the cross. So I'm going to attempt to share another solution of a genetic cross showing sex-linked diagrams in our next segment. For now, I'm going to hand you over to Looney.
Thanks, Eugen. Guys, we're going to take a quick break, but before we do, don't forget to send us your questions and comments on our WhatsApp line, as well as on all our social media platforms. We'll see you straight after this. Welcome back from the break, guys. Thank you so much for tuning in. Don't forget to enter that competition and stand yourself a chance to win 2 gigs of data. Enough talking, over to Eugen.
Welcome, guys. It's been 2 long sessions. We're going to stick it out. We're going to get into a session now where we're going to look at sex-linked inheritance, and we're going to spend some time looking at human evolution, take a few questions on that. I've got some friends here that I'm going to introduce to you in a bit, and we're going to chat about their characteristics and how they will help us in trying to understand the development of humans over a period of time. So let's get back to sex-linked inheritance, and then we'll move swiftly onto human evolution. So guys, as I mentioned before we got into the break, that there are questions that we call in genetics that contain examples of the inheritance of sex-linked characteristics. And remember, we mean that these genes are found on the gonosomes. And by that I mean the gender-determining chromosomes, which are either the X or the Y chromosome. And often we've studied examples of X-linked inheritance, and hence we call that the inheritance of sex-linked characteristics. An example of a sex-linked characteristic is hemophilia. Other examples include color blindness. So here is a sex-linked disease, and it is a recessive condition, meaning that individuals that are affected will have the recessive allele. As I mentioned, when we indicate the inheritance of sex-linked characteristics, we've got to show the gender, and we know that the gender of individuals are indicated using the letters X and Y, and in this case, females have 2 X chromosomes and males have an X and a Y. We also know that we've got to indicate the inheritance of characteristics using alleles, and in this case, we've got to use them as a superscript. And in my instance, I've created here an individual that has the dominant allele and the recessive allele, and that is a female who we refer to as a carrier or a heterozygous individual, meaning having the dominant allele and the recessive allele. And here's a male, and a male could either be dominant, meaning not affected, or could be affected, and in this case having the recessive allele. So let's look at this question. A normal father, meaning unaffected, and in genetics we tend to refer to normal as individuals that are not affected by a specific genetic condition. So a normal father and a heterozygous normal mother, meaning that this female is a female that has the dominant allele as well as the recessive allele, have children, represent the genetic cross between the two to show the possible genotypes and phenotypes of their children. So let's look at how this genetic diagram is laid out. As it is a genetic cross using sex-linked inheritance, we've got to indicate the genotypes and the phenotypes using the chromosomes X, Y, and XX, and that's just my reference point for now. And we know that the dominant allele in this case is H for individuals that are not affected, and the recessive, in this case small h, refers to individuals that are affected with the condition. So these individuals have been affected, in this case, with hemophilia, or maybe if we're looking at color blindness, these would be colorblind individuals. Right, so the phenotype, we start off laying our diagram with describing the phenotype of the individual. So a normal father, meaning the individual is not affected, has obviously married or has children with an individual, a female who is a carrier. She is not affected, but she is a carrier, meaning that she has the dominant allele and recessive allele. The male genotype must be indicated using the X and the Y chromosome, along with the allele, in this case, the dominant allele for the condition of hemophilia. In our third step, we've got to show the formation of gametes, and this is where we separate out the X and the Ys, and along with the X, so do we keep the allele for the condition, and in this case, the dominant allele and the Y. And what's very important to note is that on the Y, there is no allele. And the reason I place emphasis on this is an X-linked condition, meaning that the gene is only on the X chromosomes and not on the Y chromosome. So it would not be found on this chromosome. It would be incorrect to indicate an allele on the Y chromosome. So we separate out the alleles, and we have, for the female, we've got an X with an H and a small h on the second X chromosome. We then show the process of fertilization, where we cross these individuals to create what we call the possible offsprings that have formed from this genetic cross. And that can be done using this cross, or if you choose to use a Punnett square, then you are able to achieve that, illustrating that using a simple Punnett square. And often learners choose to use either illustration, and I think there are preferences in the way you've been taught this section, and you might be comfortable using the cross diagram, which I've indicated above, or you may choose to use a Punnett square, which I've indicated here. And a Punnett square is essentially a set of squares that are used to work out the possible offsprings. And hence you can simply cross the gender first, so that's XX, and we cross that and we will get this individual. So this is a cross between individuals that are in this block and in that block. We also can cross this block, which is a cross between individuals here, or gametes here and there, and you would get this possible allele or genotype. We then cross the last row, and that's going to give you a dominant allele as well as the recessive allele from this individual here. Okay, that goes in there. And in the last square we have an XY, that's a male, and we see that the recessive allele comes in from there. So when we look at the genotype ratio, it is an expression of the possibilities of the offsprings that might be produced. And we then end off the question looking at how they would physically appear, and in this case we describe this as two normal females or daughters with one normal or unaffected male, with the possibility of also having one son that might be a carrier. So this shows you the possibilities of their offsprings. And if I were to ask you a question, what is the possibility of this couple having a son that is affected? So we will need to focus on just the sons, and when we look at the sons, there is a possibility of having a son that is normal and a son that is either affected with this condition, hemophilia, or let's say color blindness. So there is a 50% chance if they have a son. But if we were to look at all the offsprings, and we're looking at all four of the offsprings, we have a 25% chance, which is a 1 in 4 of all the offsprings having the condition, which in this case is this son that might have that opportunity. So guys, we've looked at the inheritance of sex-linked characteristics. As I mentioned, we need to get into evolution, and let's look at that. So At the start of evolution, it's important for us to be able to understand what are the similarities between humans and what we call the African apes. And I think I've exhausted time on looking at the inheritance of characteristics, but there is a special emphasis that you need to place on this section of evolution. And I'm going to try and do that now. So when we look at the similarities that humans share with African apes, I've used a very schematic diagram to represent this. And here you can see that humans and the African apes have a large brain. Their eyes are present in front, and by that I mean they stick immediately in front, and there's clear stereoscopic vision and the ability to be able to look straight ahead. We have freely rotating arms, and you're able to rotate your arms and move them. We have a long upper arm, and we have the ability to rotate the elbow at the joint. We also have fingertips which are covered with nails instead of being covered with claws. Okay? You also see that we've got an opposable thumb, and that gives us the ability to be able to power grip as well as precision grip. We have an upright posture, meaning that we stand upright, or in other words, we tend to be bipedal, and that's a feature that is similar to most African apes and humans. Okay. What's also important is for us to be able to find the differences between humans and African apes, and I've tabulated that. And if we were to compare that, I've got some friends, as I mentioned, so I'm going to go through this and very quickly refer to these. So when we go through a human skull, so this is a modern human skull, you will see there's a distinct difference between this and an African ape. And here I've got a gorilla, which is clearly, from a size perspective, distinctly larger. We also notice the development of significant cranial crest and well-developed brow ridges. We see that significantly reduced to having a smoother cranium and even the absence of developed brow ridges. When we compare the shapes of the face, we see that the shapes of the teeth are significantly different. We know that one has a significantly protruding lower jaw, whereas the human, the more modern face is much flatter. We see the reduction in the cheekbone. We see a distinct, well-developed cheekbone in your greater African apes. We know that the canines are larger, and that indicates the type of nutrition they have, whereas in humans we see a significantly reduced size of dentition. We see that the canines are significantly smaller. When we compare the shape of the jaws and the palate, we see a significant difference in the humans being much rounder and more subtle, whereas the gorilla having a distinct rectangular and much more robust palate and jawline. So let's get back into these differences. As I mentioned, we can compare the cranium, we can compare the brow ridges in terms of being defined and well developed in the African apes. We also can look at the spinal cord. In humans, it's an S-shaped spinal cord. When we look at the African apes, they have a C-shaped spinal cord. If we were to compare the pelvis or the pelvic girdle, humans have a short but a much wider pelvis. And if we were to compare that to the African apes, they have a narrow and a much elongated pelvic girdle. Let's move on to other features that differentiate humans and African apes. So we can look at the canines. They are much smaller, whereas African apes, as I mentioned, much larger. The jaws, again, much smaller, much larger. We spoke about the protruding lower jaw, and we referred to that as prognathus. So humans have a less prognatism, whereas your African apes show significantly more protruding lower jaws. The foramen magnum is much more towards the base and the center, indicating a distinct upright and bipedal posture. Whereas if we compare that to the foramen magnum of the African apes, we would see that, and from this you can see it's much more towards the rear end, indicating a possibility of being able to stand upright but not significantly bipedal. We're going to look at a few more differences, and here's a question which I'd like to end off with. I'm going to read through the question and I'm going to try and give you an overview of how to explain this. Study the two skulls shown in the figure below and answer the questions. And when you get a diagram like this, it's important that you look at the features on the diagram and try and establish what is the difference between them. And if you compare them, you can clearly see the skull of A seems more human-like, and you would know that a human-like skull shows a significant development of the brain. If we compare that to skull B, you can see significantly smaller development of the brain. Let's move on to the question, and we would try and answer this before we end the session. Which skull, A or B, is that of a non-human primate? And guys, if you were to compare these, I mean, you could clearly see the distinct feature of a large brain, a flat face, forward protruding eyes, smaller teeth, that all are indicative of a human skull. So the non-human skull has a significantly smaller brain, it's got distinct brow ridges, and it has much larger teeth. And essentially, for 1 mark, you should be able to easily give those features. I'm going to try and get to one more question and just give you an overview of this before we wrap up. List 5 observable reasons based only on the features that are visible in the diagram for your answer. So guys, in this question, you've got to be able to list the 5 observable reasons. And often we get learners that provide reasons that are not evident from the diagram. And hence, it's important that you refer to these diagrams and look for differences or similarities, and you can easily identify them by looking at these images. Guys, that's a wrap for me. We've exhausted the section on evolution. I had the opportunity to show you some features, but I'd like to end the session and wish you well for paper 2. Over to you.
Matrix, it's been an amazing show as always. Thank you so much for joining us. And a big congratulations to all of our competition winners who will be announced after the show on Facebook. To check out more revision schedules, please go to www.wazamatrix.co.za. From me, Luni, Nicolene, and Eugen, it's thank you and goodbye.